So this is part 4. We will be seeing in this part the DNA-seq protocol, and it would be an overview of the first part, which is the library generation. So you have here a very general protocol and simplified protocol to generate libraries. So the general protocol starts with a DNA purification, then fragmentation of the DNA, then what we call polishing the end, which is a repair to the end and a process of A-tailing. We will be seeing the details of these. Then you ligate your adapters, you size select, and you perform a PCR amplification before sequencing. So in fact, there are several kits that are available according to the need that you have, according to your input DNA. So depending on your experimental procedure, you can buy these kits. So this is usually done by the platforms. There are kits that are compatible with Illumina. There are others that are compatible with Ion Torrent. You have here just some examples of the ones that are used for Illumina. But you can have a lot more on internet. So the first thing to do, so step 1, is to purify your DNA. Now you need also to check the quality of your starting material and to check its quantity, of course. So determining the quality and the quantity is important for the next steps of the analysis, like size selection, for example, because these steps would depend on that amount of starting material. Anyway, your goal through the whole process would be to ensure that you do not bias your library construction by any means. So people used to check the quality on a gel, but now you would more often use a bioanalyzer that is developed by Agilent, for example, or Biorad companies. And then you also need at this stage to make sure that you properly design your experiment. Oh, sorry, I forgot the quantity control, which is done by a nanodrop or a qubit. I'm sure that you have these technologies in your different labs or that you heard about them. So then you need to make sure that you fully think about the experimental design of library construction. So would you be using single read sequencing or paired end sequencing? Would you be multiplexing or not? Are you having a genome for which you have a reference, or you don't have that kind of genome and you would like to perform de novo sequencing, etc., etc. So there are a lot of considerations to take into account. So first one is the sequencing by single reads or paired end. So basically, single reads is sequencing from one end, whereas with the paired end you sequence from both sides, as you can see it here. And this usually helps to discover, so using the paired end usually helps to discover insertions or deletions in the genome, and we'll be seeing this a little bit later. So would you be also multiplexing or not? So this multiplexing or not would depend on the coverage you want for your sequencing, meaning how many sequencing you would like to have for each of your samples. So for example, if someone tells you that you can sequence 1 million reads in one line of a flow cell, if you remember, we saw on a flow cell 8 different lines. So if this is too much for you, for the coverage that you want, and let's say that 50 million reads would be enough for you, then you can use barcodes to sequence 2 libraries in one line, and this reduces the cost, basically. So the principle is simple. You have here an example that is taken from Illumina, the Illumina website, where you have 2 DNA samples that are multiplexed. So this means that they are attached to a specific barcode sequence that allows you to identify the sample from which it is originating. The libraries are then pulled together and sequenced in parallel, so in the same line of a flow cell. And you can see it here. You have amplified reads that contain fragments and barcode sequences. And reads from each library would be differentiated by using that same barcode in order to demultiplex and come back to the sequences of origin, and then you can go back to your reference and map your reads on it. So step 2 would be to fragment your DNA. So the degree of fragmentation of DNA is controlled by the time of fragmentation and a temperature, for example. So there are several key steps to take into account. Both factors, so time and temperature, for example, are factors that may be modulated to achieve the desired fragmentation output. This is especially true for small genomes, like viral genomes, for example. And for this sample, these kind of samples, the fragmentation time may be reduced. And this makes indispensable, I would say, to control the reaction time and temperature. You also need to keep in mind that the proportion of fragmented DNA that is successfully converted to adapter ligated molecules will decrease as the input is reduced. So you need to make sure that you have enough input material that is ideally of a high quality. So you need to make sure that you controlled for all these in the previous steps. Now step 3 would be to repair the ends. So basically you have an overview of what is happening here. So basically at this stage you would convert overhangs that are here. You would convert these into blunt ends. And then you would phosphorylate the 5' end. So this is the first step, the end repairing. Then you would add A base to the 3' end of the phosphorylated DNA fragments. So this is appearing here as an addition of A overhangs. Then step 4 would be to attach adapters to both ends of fragmented DNA, and these adapters can be provided with the kit, so classical ones, or you can choose to design your own if you want to target specific regions of interest. And you have to note that the adapter concentration affects ligation efficiency, as well as the adapter and adapter dimer carryover. So the ligation efficiency is quite robust for adapter to insert molar ratios between 10:1 and 200:1. And you have to use higher adapter ratios if you have low input applications. You need to make sure that you always use high quality adapters if you design them by yourself, because this could impact later stages. So make sure that you have adapter quality control. Then you need a step that is called post-ligation cleanup. And this is really important because it allows you to remove unligated adapters and eventually the adapter dimer molecules from the library before going into the amplification stages. So size selection is also important. Why? Because this serves as kind of an internal control to ensure that each library would get similar reads or coverage. So, for example, when libraries are loaded into the flow cell in order for you to make the cluster generation, the samples that would contain lower molecular weight DNA or DNA of smaller length would be preferentially amplified via clustering, and this would introduce a bias in your library. And you have also a problem here at that stage of material loss. So it is something you need to control because size selection would inevitably lead to a loss of sample material. So you need to focus on these points. So size selection is performed thanks to the use of specific beads that are called SPRI beads. And in fact, we use here a differential concentration of these beads to keep some sizes and release the others in the supernatant. And then, as you can see it here, the size ranges in the gel according to those that would be kept on the beads and those that would be kept in the supernatant. So the selection is done simply by playing with the fragment sizes and doing SPRI or reverse SPRI. So step 5 would be to amplify your library. So once you have your library workflow that is optimized and you went through the whole workflow and you consistently yielded the amount of amplified library of the required size, so that you size selected, a quantification at this stage would allow you, so this is what is noted here, QC, would allow you to assess the efficiency of two separate things. First, the library construction itself, and then the library amplification. So the protocol can be used for whole exome sequencing, for whole genome or for whole exome, but also for target regions. So it is also possible to choose or customize your regions of interest by using that the same method that is used for whole exome, so targeting specific regions, or by PCR amplifying a target of interest. The choice would essentially depend on the biological question you have. Now, once the reads are sequenced, you have a quality control to perform in order to ensure that there was no cluster amplification bias and that no bias would be, you have no bias that would be related to a flow cell effect or a line effect or a cluster effect. And the output would be a file containing the sequence of the millions of reads that you obtained. And we will be seeing how these files are analyzed in the next part.